Assay is the amount of API or amount of the active or amount of the drug substance present in the formulation. Sometimes it is also called as potency by some of the professionals. The assay limits are generally 90 to 110% of the label claim, and sometimes these are the values like 95 to 105%. Generally for the US market, the assay value is required to be within 90 to 110%, and for Europe market generally it is 95 to 105%. If there is failure in the assay, then it will trigger the OOS, that is out of specification, and once it triggers the OOS, then investigation is required to be carried out. As the assay has a limit or range, for example 90 to 110 or 95 to 105% of the label claim, the failure may occur at both ends. The failure may be if the assay is below the lower limit or it is higher than the higher limit. If it is higher than the higher limit, that time different investigation will be there, and if it is lower, then different investigation will be there, because the reasons for higher and lower assay reasons will be different. This video is specifically for the assay failure due to degradation. Have you ever come across a situation where you found that the assay is on the lower side? That may be the process loss, that may be the degradation, that may be the problem in the analytical testing. I welcome all the viewers to this video on out of specification investigation, OS investigation for the assay failure. So let's start with the introductory part. Out of specification for the assay failure, we need to understand the causes, phases of investigation, and what the corrective actions are required to be taken. So when the assay fail because of the percent of the active ingredient content falls below the acceptance limit, that indicate the loss of potency due to the degradation, or it may be due to the process loss. There are various reasons, and in this video I am considering the loss of potency due to degradation. Assay or label claim is important because it directly has impact onto the product efficacy. It has impact onto the patient safety, and it has impact onto the quality compliance, regulatory compliance. If the assay limits are not in the range as per the quality requirements, then it will lead to a regulatory non-compliance, and that's why the OS investigation is to be carried out and documented. Now, if you consider the loss of the assay due to degradation, loss of the API, loss of the drug substance, or lower side assay because of degradation, then we need to understand what are the reasons. So first is oxidation, because for the degradation, because of the hydrolysis degradation can happen, photo degradation, thermal degradation, because of any interaction with the excipients, packaging defects, improper storage, anything can happen. So we need to consider all these reasons. Examples like vitamins are there which undergo oxidation. Hydrolysis can happen to the aspirin, photodegradation to different excipients and APIs may happen. Thermal degradation may happen to the proteins and amino acids. If you observe the lower side values for the assay because of degradation, then there will be a investigation, there will be a OS login and investigation will be started. So in first phase, initial laboratory investigation will be there to check the analyst error. So correct the sample preparation, dilutions, weighing, cross check the chromatograms so that we can find out is there any problem because of the analyst or analyst error. Then because of the instrument calibration, method verification, HPLC calibration, column performance, mobile phase check, different things are there which are required to be checked. We need to confirm the method suitability. Re-injection retest can be carried out to rule out the problem or error in the sample preparation or handling of the standard and the samples. Then there will be hypothesis testing, comparing the impurity profile with known degradation products, so that directly you can understand whether the assay is lower side because of the degradation or it is because of the process loss. So then we need to evaluate against the stability study results. Phase 2 investigation will be the extended investigation. In this, all the things will be evaluated, like manufacturing review will be there, storage and distribution review, and checkpoints will be there, packaging review will be there, and data correlation will be there. Manufacturing review will involve deviations in the blending, compression, granulation, drying, overheating due to processing, and many more, depending upon the manufacturing process and the specific formulation. Storage and distribution checks will be there for the environmental excursions, like excursion increase in the temperature, humidity, and cold chain integrity. Packaging review will be there, considering the container closure system integrity, leakage, improper sealing, or material incompatibility. Data correlation will involve cross-check with the long-term stability data, accelerated stability data, retained sample analysis. It will help to differentiate the real degradation versus the analytical anomaly. That means you need to understand and investigate whether the assay is lower side because of the degradation, or it is because of the analytical error, or it is because of the process loss. Then once you get the root cause, you need to go for the kappa, that is corrective and preventive actions. If root cause is found, that time you need to go through the series of actions to go to the effective kappa. Revise the storage and handling SOPs, strengthen the packaging system. You can add antioxidant stabilizers, improve the warehouse monitoring, train the people, operator, analyst, so that they can avoid such instances. Now you might be thinking that at which stage it will be done. That means whether it is in the manufacturing batch, at plant level, or at manufacturing site level, or it is in the R&D. So whenever it occurs, the formal investigation is required to be carried out. But if it occurs at the manufacturing site and not in the research phase, that time it will trigger the OS and kappa will be there. If it happens in the R&D, then no need to go for the OS. It will be a formal investigation and understanding about the batch, understanding about the composition and process because of which the degradation happened. And if it happens in the research phase, then proper care to be taken in the next trials. But if it happens in the manufacturing site, OS will be triggered. SOPs are required to be revised based on the kappa. Different things will happen to support the kappa system. Regulatory perspective for the OS. So OS investigation must follow the guidelines like 21 CFR or country specific guidelines, guidelines provided by the EMA, ANVISA, Swissmedic, MHRA, and many more. Then documentation should be there, which is required to be complete, scientifically justified, and it is reviewed by the regulatory affairs and quality assurance. So regulators expect the root cause identification, and based on that the kappa should be there, and kappa effectiveness is required to be checked. So this is regarding the OS in the assay. OS because of the assay failure because of degradation. So it is a failure. Degradation pathway, degradation condition should be understood and investigated, and based on that, the corrective and preventive action should be taken. All the kappa activity will be based on the root cause for the lower side assay. Thank you for watching the video. Let's meet soon with another interesting and informative video. Thank you.